Hello everyone, welcome to another episode of Tony's Insights. Today's video is on urea cycle. Let's dive right in. The urea cycle. What is urea? What is urea? So urea is the urine you excrete. The major component of the urine you excrete is actually urea. Like to be more specific, 90% of nitrogen containing components of urine is urea. So let me draw the chemical structure of urea. This is So as you can see, there are two amino groups in urea. So these amino groups, when they are not reused for the synthesis of new amino acids or other nitrogenous products, they will then be channeled into the production of this urea through the urea cycle. When they are then synthesized through the urea cycle, they will then enter through the bloodstream, they will pass to the kidney and then be excreted in urine. So this is the chemical structure of urea. So there are things that make urea to be an ideal nitrogenous end product. There's things that makes urea to be so ideal as a nitrogenous excretory product. One of them is that urea is highly soluble in water. And as urea is highly soluble in water, it can then be excreted, urea. And then as it's highly soluble in water, it can then be excreted in large amounts in urine. Two is that it is non-toxic. As it's non-toxic, it's not harmful to the body. The third one then is that first one, the fact that highly soluble. It is highly soluble in water. It dissolves in water. It can pass through the urine. Then second one is the fact that it's non-toxic. Non-toxic. And third one is that it is metabolically inert. When I say that metabolically inert, it means that it won't start entering another metabolic pathway or start acting upon. It won't start reacting with other substrates. It just passes through the urine. So the third one is that it is metabolically inert. Yes. So let's look at the urea cycle. Now, the urea cycle occurs in the liver mitochondria. The first two steps of the urea cycle occurs in the liver mitochondria. The urea cycle occurs in the liver. So let's look at the urea cycle. Okay, so like I said, urea is the major disposal form of amino groups that are derived from amino acid. So let's look at the urea cycle. So the urea cycle starts with CO2 plus ammonia. So do you see this ammonia now? It is gotten from the amino group of the amino acids. It is gotten from, for instance, let's say glutamate. Glutamate is an amino acid. So when glutamate is an amino acid contains an amino group. So when the amino group has been removed from the amino acid, you call it deamination. So deamination of amino acids, that's where this ammonia comes from. The other places the ammonia can come from, in our diet, for instance. This ammonia can come from our diet or various other sources in the body. So, but the primary source is from deamination of amino acids, for instance, glutamate. So, this ammonia now, and let me see, the first two reactions of urea cycle occurs in the mitochondria. So, right now we are in the mitochondria. We're in the mitochondria, right? This is we're in the mitochondria. So, like I said, this amino group now came from the deamination of the amino acid. Then the CO2. This CO2 comes from citric acid cycle. You can watch my video on that, citric acid cycle. So this CO2 came from there. So the CO2 will combine with this ammonia to form a compound called carbomoyl phosphate. It will synthesize a compound called carbomoyl phosphate, and it requires two molecules of ATP. ATP, that's the energy form of an energy form. Two molecules of ATP is required for the synthesis of carbomoyl phosphate. Now, and then it will give out two molecules of adenosine diphosphate plus one phosphate group. You might be like, where is the second phosphate group? Because if two ATP were to cleave, it should give two ADP plus two inorganic phosphate, two molecules of inorganic phosphate. The second phosphate group will be incorporated into the carbomoyl phosphate. So let's look at the structure of the carbomoyl phosphate. So this is the structure of carbomoyl phosphate. This part now is gotten from here. Let's look at it. This part here, it is gotten from this part here, and then this part here is gotten from here, and then this phosphate now, it is that inorganic phosphate I was telling you about, the inorganic phosphate from the hydrolysis of, sorry, from the cleavage of the 2-ATP. So the enzyme that synthesizes this is called carbamoyl. Look at the name. So this is carbamoyl. So this is carbamoyl phosphate. Sorry, this is carbamoyl phosphate. And what do you think would be the name of the enzyme that catalyzes this step? The synthesis of carbamoyl phosphate. So the enzyme that catalyzes is called carbamoyl phosphate synthetase. That's carbamoyl phosphate synthetase 1. This is called 1. This one is called, that's actually a carbamoyl phosphate synthetase 2. That's a carbamoyl phosphate synthetase 2. So because of the carbamoyl phosphate synthetase 2 and the pyrimidine nucleotide biosynthesis. So the one that catalyzes this step is the carbamoyl phosphate synthetase 1. So let's look at the second step of the reaction. For the sake of the carbamoyl phosphate synthetase 1. Yeah. So the next step of the reaction now is the formation of citrulline. So see what happens here. Citrulline will come in. Let's say like this. Let's say it's going like this. It's going like this, right? So next, what will come in? What ornithine will come in? A molecule of ornithine will come in to form citrulline. So ornithine will come in plus carbamoyl phosphate to form citrulline. So it might be like, where is this molecule of ornithine coming from? Ornithine is coming from regeneration. When I say regeneration, I mean each turn of the cycle, ornithine is regenerated. Like with each turn of the cycle, like by the time we go through this cycle now, we'll reform ornithine that will go through the cycle again, like so it's constantly regenerated. So let's look at ornithine, the structure of ornithine. So this is it. This is the structure of ornithine with the amino group here. Then you can either write it as like the CH2 bracket 3 to show that there's three methyl groups, or you can decide to just write everything out. You can decide to kind of show it out. CH2. Let me just make it here, the amino group here. So this right now is ornithine. Yeah, this right here is ornithine. So see what happens here. Ornithine will come and join with this. Ornithine will come and join with this to form citrulline. So what actually happens is that the carbamoyl portion, this is the carbamoyl portion of the carbamoyl phosphate. This part now will actually join with this part, with the removal of this part as inorganic phosphate. Do you understand? So this part now, this is the inorganic phosphate. This is the structure of inorganic phosphate. So it is removed out. This one goes out. The carbamoyl portion will stay. It will come and join with this to form this. So that's for the next step of this second step. Okay, so writing it out to now be, we write all these things. COO minus CNH. So here's what happens. This part now will go on top of here. Yes, it will go on top of here. So this right here, that's what we call the citrulline. The citrulline. What do you think would be the name of the enzyme that transfers the carbamoyl group from carbamoyl phosphate to ornithine? The enzyme will be ornithine transcarbamylase, OTC. Yeah, OTC. Ornithine, sorry. It will be The name of the enzyme will be ornithine transcarbamylase, because there was a transfer of the carbamoyl group from carbamoyl phosphate to the ornithine to form citrulline. Ornithine transcarbamylase here, or we call it OTC for short. This one I can call it CPS1 for short. Okay, so now the next step of the pathway is the formation of adenosine. Adenosine is formed in the next step of the pathway. So see how it happens here. Aspartate will come in. You understand? Aspartate will come in. So look at how aspartate comes into the cycle. So aspartate will come and merge with citrulline to form adenosine. And that's where aspartate is an amino acid, right? Aspartate comes to donate the second amino group. This is the first. You know that I thought that urea has 2 amino groups. This is the first amino group. This is now the second, the one I want to show now, for joining of aspartate. So see what happens here. See, yeah. So those kind of yes, positive. Hope you are seeing this. If you are not, please, there is a demarcation. This part is for the ornithine. This part is for the so okay. So this is the aspartate. This is called aspartate, an amino acid, a 4-carbon amino acid. So aspartate, what aspartate is coming to do is to donate this amino group. To donate this amino, that's just what aspartate is going to do. The second amino group of urea is gotten from aspartate. So aspartate will come in. Okay, let's look at it. Aspartate will come in, it will join. So let's see how it joins. It's a very, very interesting. Okay. I'm just redrawing this. I'm redrawing this structure. Then I'll then show you where I'm redrawing this structure, and then I'll show you where this aspartate comes in here. So, okay. Sorry. Okay. So, CH2 here, CH2, and then this one came in, NH, and then carbon 2, NH2. But see the thing here now. The aspartate will come in, the amino group will come, and this amino group will come and join at this carbon here, removing this one here. So look at how it happens. It will just kind of join here. Yes, it will, okay. It will join here. So this is now NH. This is now NH from this, and then combine with carbon to hydrogen to CH2. So this right here, this entire, but now see this, see the problem here. Carbon is doubly bonded to oxygen, right? Carbon has to be bonded to 4 different atoms. 1, 2, 3, and 4. 1, 2, 3, and 4. But here now, as you have removed this double bond that holds the oxygen, as you've removed everything, as you've removed this out now, if you count it now, it's now bonded to 1, 2, and 3. So therefore, this is the nitrogen, the ammonia group. So therefore, you have to add a double bond here. You understand? You have to add a double bond here so that carbon will now be bonded to 1, 2, 3, and 4. This nitrogen has to be bonded. Nitrogen, in chemical reaction, nitrogen is seeking to bond to 3 different atoms, or is seeking to form 3 different bonds. So there's 1 here, 2 here, and there's a 1 with the hydrogen here. So you understand? So this whole thing is now the structure of adenosine. What do you think will be the name of the enzyme that forms adenosine? What do you think will be the name of the enzyme that synthesizes adenosine? The name of the enzyme is adenosine synthetase. Synthetase. For my previous videos, you would have known that as I said synthetase, you should have been like, where is that molecule of ATP? Actually, there is a molecule of ATP that came in. Yes. ATP also came in. A molecule of ATP came in with the So this reaction is ATP dependent. So as I was saying, the name of this enzyme, is adenosicinate synthetase. It catalyzes the formation of adenosicinate from citrulline and aspartate. You understand? So, sorry. Okay, so it is adenosicinate synthetase. Yes. So let us look at the next step of the reaction. Okay, so I was saying, the next step of the pathway. Oh, I actually forget to mention something very critical. Like I told you that the first two reactions of the urea cycle occurs in the mitochondria. That's this one and this one. After this citrulline is formed, it will then enter into the cytosol. It will enter into the cytosol that's outside the mitochondria. So this part from here now, all these parts occurred in the mitochondrial matrix because that's where the enzymes of these first two steps are formed. But after the first two steps, other enzymes are then found outside the mitochondria and the cytosol. Yes. So actually, this is ATP we cleave to AMP and then pyrophosphate. This is a sorry. ATP cleave to form AMP and a pyrophosphate. So, okay, and remember this is the final ATP that is used in this cycle. This is where the first two ATP was used. This is now the final ATP. Now to the next step of the reaction, we'll now be looking at, do you remember that I told you that all aspartate came to do was to donate this amino group. It didn't come to donate its entire self. So it wants to donate this amino group and then the carbon skeleton will leave. So the next step of the pathway is showing when the carbon skeleton leaves. That's this part here. There will be a splitting at here. This part will now leave. So that's what we are looking at in the next step. So here is the next step. Okay, make it longer. Here is the next step. So what we will leave is this part here, leaving behind this amino group because it has not donated the amino group. So now looking at it now, this is it. Sorry. Yeah. Yeah. So this now is fumarate. Fumarate is when you now remove the amino group from aspartate, you form fumarate. So what is now remaining is this entire thing here. And this entire thing here now is what we call arginine. Okay. So this is it. Drawing the structure of this now is this is it. And then you draw. So and then okay, that's three this thing, and then you then go on to put the ammonia, the amino group with this carbon. Sorry, this is for the sake of, let me just put this down to give the NH, and then the carbon going to the N, and then this one right here, the NH2 here. So this right here is called arginine. And what do you think would be the name of the enzyme that splits adenosicinate? Remember this enzyme was called adenosicinate synthetase, the one that forms synthetase. The next one that will now be called, this one here will be called, the one that lies this step will be called adenosicinate lyase. Lyase means splitting. Enzymes that catalyze splitting of compounds, like it's called a lyase. So adenosicinate lyase is a group of enzymes that catalyzes the splitting of. So actually now this is adenosicinate lyase. This is adenosicinate lyase. So okay, so that's the step of the reaction. So the final step of the reaction is where the urea is produced. From here now, from this arginine now, it will form ornithine as well as urea. Urea will leave at this point. Yes, urea will leave at this point. At this point, urea will leave, And remember when I told you, so this, remember, this is the structure of urea. This right here is urea. So urea will leave with the formation of ornithine. Understand? So the ornithine will go back and for another turn of the cycle to reproduce urea again, and so on and so forth. Remember when I initially told you that ornithine is regenerated in this cycle. So ornithine will go back with this and continue forming it. So this right here is the urea. And the name of the enzyme that catalyzes this reaction is arginase. Arginase. Okay, arginase here. Arginase. The breaking down. So it was the breakdown was at this point here. Yes, at this point here. So and the breakdown required H. It required H2O. So this what actually happened in this reaction is the hydrolysis of arginine to form ornithine and then urea. You can see that H2O now, the H2O now donated the oxygen that we see here. So this is the structure of urea cycle. This is the structure of urea cycle. Let me just quickly go through the video one more time. So what let's look at starting from here. I thought that the amino group was gotten from deamination. Amino group was gotten from deamination. That's removal of the amino group from the amino acid. This amino group was gotten from there. This CO2 now is actually from citric acid cycle. When you look at my video on citric acid cycle, you see how CO2 is produced. CO2 combines with this one plus a phosphate group from ATP to form carbamoyl phosphate. And an enzyme that catalyzes synthesis of carbamoyl phosphate is carbamoyl phosphate synthetase 1, because there's actually a carbamoyl phosphate synthetase 2 that catalyzes another reaction. So this one now will then go on to form citrulline. It will go on to form citrulline. There will be a cleavage, like the phosphate group will leave. See it here, the phosphate group left. See the phosphate group here, it's left. So this one now will come. So there was, at this type of reaction, there was a donation of the carbamoyl portion of the carbamoyl phosphate to ornithine to form what? To form citrulline, yes. So you can see it here. This one donated on top of here to form this. So the next step was now synthetase. Aspartate came in to join with this one to combine with citrulline to form adenosine. The reason is combining so that it can for the amino group to donate the amino group. And when it has donated the amino group, then there will, when like it has come in, it will then split so that the carbon skeleton will leave and only the amino group will remain. So see it here. So see it here. So now the next step of the reaction, the final step, is the formation of the urea, the whole point of this cycle, the formation of the urea. So we split it at this point, and this was the urea leaving. The urea left. The urea actually, so the fate of the urea is that as the urea is formed, it will then enter the bloodstream. From the bloodstream, it will then enter the kidney to be excreted as urine. So this is it for urea cycle. I hope you enjoyed this video. If you do, please subscribe, like, and comment. Thank you very much.